Welcome to Catch This. My name is Trudy Ray. You know norovirus, that nasty virus that is typically associated with outbreaks on cruise ships? In this video, I describe a new study that shows that this virus could potentially be transmitted through saliva, not just by the traditional fecal-oral route. Please stay tuned. Norovirus and rotavirus are considered to be enteric pathogens because they are traditionally thought to be transmitted by the fecal-oral route. In other words, when consuming food prepared by someone who did not wash their hands properly after using the bathroom. Unlike rabies virus, which replicates in the salivary glands and transmits through saliva, the presence of noroviruses and rotaviruses in saliva is usually considered to result from contamination, for example by vomitus or acid reflux. It was therefore surprising when a recent study showed that neonatal mice, referred to as pups, can apparently transmit enteric viruses to their mothers during suckling, following oral inoculation with mouse norovirus 1, or MNV1, or epizootic diarrhea of infant mice. Let's just call it EDIM. It's a mouse rotavirus. Pups had secretory IgA in their intestines that increased gradually over the course of 2 weeks. Secretory IgA is a type of antibody that provides immunity in mucous membranes, such as those of the mouth, nose, and gut, and it is typically passed from mothers to infants during suckling. Interestingly, this secretory IgA increase in pups correlated with a similar increase in the secretory IgA in the milk of the mothers, even though the mothers were not infected and did not have any antibodies to either virus at the start of the experiment, meaning that they were seronegative. Considering that pups can't produce their own secretory IgA, it is likely that the pups infected the mothers during lactation, who then produced the secretory IgA and passed it back to the pups through their milk. To rule out the possibility that the mothers became infected by consuming feces-contaminated food, in other words, by the traditional fecal-oral route, because they shared a living area with their infected pups, the authors of the study orally inoculated pup-free seronegative mothers with EDIM. Then they treated them with oxytocin to induce milk production and analyzed the milk for secretory IgA and their mammary glands for viral RNA. Although the mothers were successfully infected, which was obvious from the presence of EDIM RNA in their small intestines, there was no detectable EDIM RNA in their mammary glands or detectable increase of secretory IgA in their milk. This result confirmed that the infected pups passed the virus to the mothers during feeding. To further substantiate this finding, the authors orally infected one set of pups, pups A, with EDIM and placed them back in the cage with their seronegative mothers, mothers A, for suckling. The next day, mothers A were replaced with mothers B from a cage of uninfected pups, pups B, and mothers A were placed with pups B. Got it? 2 days after this switch, all mice were euthanized and analyzed for the presence of viral RNA. All mothers had EDIM RNA in their mammary glands, suggesting that both sets of mothers became infected by suckling pups A. And all pups had EDIM RNA in their small intestines, suggesting that pups B became infected by feeding from mothers A or by ingesting their fecal matter. The next set of experiments aimed to determine whether saliva contains enteric viruses and if it could serve as a means for transmission. Infection of adult mice with MNV1 or EDIM revealed that these mice produce and shed virus in their saliva for up to 10 days post-infection. To see whether the saliva was infectious, the authors fed it to pups as a means of inoculation. At 3 days after infection, the pups had significantly high viral genome levels in their intestines, comparable to those observed in pups inoculated with virus of fecal origin. This result suggested that the viruses replicated in the intestines and confirmed that saliva can be a conduit for transmission of these enteric viruses. Next, the authors wanted to see whether these viruses replicate in the salivary glands, so they orally infected pups and adults with with various strains of norovirus and then isolated their submandibular glands, the largest component of the salivary gland complex. They then measured the number of infectious viral particles inside these glands using a TCID50 assay. This is an alternative to the classical plaque assay used in virology. The TCID50 assay quantifies the amount of virus needed to infect 50% of cells in culture. The authors found that each of the viruses increased in quantity by about 100,000-fold throughout the following 3 weeks compared to the input level, suggesting that noroviruses do replicate in the salivary glands. When they treated the mice with a drug that inhibits the norovirus polymerase enzyme, there was a decline of virus in the salivary glands, suggesting that this enzyme inhibited viral replication and confirming that replication occurred inside the salivary glands. TCID50 analysis of various cell populations isolated from the submandibular gland revealed that MNV1 replicates in epithelial and immune cells, both of which express CD300LF, the gene that encodes the intestinal receptor for all known strains of mouse norovirus. MNV1 infection of mice lacking this CD300LF gene led to no detectable MNV1 replication in the salivary glands, suggesting that this receptor is needed for infection of submandibular gland cells. When the authors partially extracted the salivary glands from adult mice before inoculation, there was a faster clearing of the intestinal infection, suggesting that the salivary glands may serve as reservoirs for replication of these viruses. The results of this study challenged the notion that noroviruses and rotaviruses transmit primarily by the fecal-oral route and raise several interesting questions. Do human noroviruses replicate in salivary glands, and do humans transmit noroviruses through saliva? Remember, these studies were done in mice. If so, would protective measures in addition to hand washing, like face masks, for example, prevent transmission of noroviruses? Are other enteric viruses, like poliovirus, also transmitted through saliva? I will be very interested in seeing future studies that address these questions. But for today, that is it. Thanks again for listening. My name is Trudy Ray, and I'll see you next time on Catch This.